So this is your second screencast for this chapter. Unfortunately, we could have like 4 hours of screencasts for this one chapter. I know I'm not going to get you to watch all of that, but we're going to go over a couple areas in detail like we have in the first and then in the second screencast here. The rest of the stuff you got to read in the book though. So do not neglect the book. You have a number of websites that are linked in Moodle for this chapter that cover other areas, like looking at the anatomy of the brain and what different areas of the brain do, stuff like that. So do not neglect the other supplemental material, including the videos. So really quickly, we need to go back to the neurons. So remember, we have a postsynaptic neuron right here and a presynaptic neuron, pre, before, post, after. So things would be traveling in this direction. There you can see that the neurotransmitters are secreted from the presynaptic neuron into the gap here. That gap is called the synapse or synaptic gap, and that is where neurotransmission occurs between two neurons. A neurotransmitter is a chemical messenger that transmits an impulse across the synapse from one neuron to another. They're contained in the presynaptic neurons in little tiny things that look like water balloons that we call synaptic vesicles that go down and bind with the cell membrane and then release the neurotransmitters into the synapse. An excitatory postsynaptic potential, or an EPSP, effects that occur when excitatory neurotransmitters cause a graded potential to occur on the dendrite or cell body of a receiving neuron. What does that mean? Well, this means that there's an excitatory neurotransmitter and it causes an increase in the electrical activity on a receiving neuron. In contrast to an inhibitory postsynaptic potential, and this is where hyperpolarization occurs, and when the neuron is hyperpolarized, it's harder for it to fire. Firing is like depolarization, and that happens when an inhibitory neurotransmitter inhibits the postsynaptic membrane of the receiving neuron. And not that you need to know this, but like glutamate would be an excitatory neurotransmitter, and GABA would be an inhibitory neurotransmitter. So let's actually look at some different neurotransmitters. But before we get there, how do we keep the supply of neurotransmitters from being exhausted? Is that what like chemical imbalances? Is that when you run out of a neurotransmitter? No. And the idea of a chemical imbalance is kind of hard to prove. I wish no one had ever used that term before. So a chemical imbalance is not something that we can measure. A chemical imbalance is more like a theory, and it's a theory that I don't even like. So you always have neurotransmitters, and they're not exhausted. I mean, if you ran out of a neurotransmitter, you would be in big trouble. So raw materials are used in the manufacturing of neurotransmitters, and they come from your food. So whether you eat meats or cheeses or pasta or potatoes, the raw materials for neurotransmitters are in food. Neurotransmitters are broken down by enzymes once they have been secreted into the synapse, and then the breakdown products are recycled for further use and sucked back into the presynaptic neuron. And sometimes the neurotransmitter itself can be retrieved intact and sucked up through a process called reuptake into the presynaptic neuron. For a substance to be considered a neurotransmitter, it has to meet the following specific criteria. It has to be contained in the axon terminal buttons. It must be released into the synapse when the neuron fires, and it must cause a postsynaptic effect after it interacts with the receptor. Here's a little drawing, and again, you can see here, this would be the action potential coming down to the terminal button of a presynaptic neuron. These are the vesicles, or little water balloons that have the neurotransmitters. Those go down, fuse with the cell membrane, release the neurotransmitter into the synapse. The neurotransmitter then binds to a receptor site. Okay, so some specific neurotransmitters now that you know a little bit about how neurotransmitters are recycled and how we classify a neurotransmitter. The first one is acetylcholine. This neurotransmitter is released from motor neurons onto muscle fibers, and it makes them contract. This is one of the very first neurotransmitters that was discovered. It's also very important in learning and memory, and it is a neurotransmitter system that is involved in disease processes, specifically like Alzheimer's disease. So some of the drugs for Alzheimer's disease increase levels of acetylcholine. Botulism, VX nerve gas, and a black widow spider's venom, they all interfere with acetylcholine transmission and produce paralysis in their victims. You might even know people that purposely get botulism toxin or Botox that produces paralysis in their face so they don't get wrinkles. Let's watch a video on nerve gas.
It's one of the deadliest chemical agents ever created. Within seconds of exposure, symptoms of nausea and convulsions take over. Without emergency treatment, paralysis, respiratory failure, and death can occur within minutes. This deadly killer is the nerve agent VX.
Weapon VX. Classification organophosphate. Lethal dose 30 micrograms.
The organophosphate type of chemicals are considered nerve agents because they attack the nervous system. The nervous system controls all the functions of the body, and so by interrupting or stimulating that, you get all these various effects.
VX in its normal state is a tasteless, odorless liquid that can be absorbed through the skin in seconds. When heated, it turns into a lingering vapor that if inhaled is even more deadly.
VX is a persistent agent, which means it's less volatile than the other nerve agents. This means that when it's exposed to the air, the material doesn't go up into the air, and it lasts much longer.
To give you an idea of how toxic the chemical is, if you pulled a penny out of your pocket, and if you looked at the penny and you looked at Lincoln's eye, it only takes a drop size of Lincoln's eye to cause lethality.
There are no confirmed cases of VX being used on people, but it's widely believed that during Saddam Hussein's chemical attack against the Kurds in 1988, VX was dispersed with deadly results. The attack on the Kurds killed over 5,000. VX is the most potent nerve agent today, but the path to its discovery dates back to before World War I.
Chemists knew that many of the chemicals used in the dye industry and other industries could be very deadly. During World War I, the Germans began to weaponize chemicals by putting in artillery shells and portable chemical cylinders and releasing them on the battlefield, and they used them very effectively.
Although chemical agents were effective in causing casualties, they were not always lethal. But that would change in the 1930s, when German scientists created the first nerve agent named Tauben. Between 1942 and 1945, they produced 12,000 tons of Tauben, as well as several thousand variations, including sarin.
The nerve agents were definitely unique compared to the earlier World War I chemical warfare agents. They had very little smell to them. They began affecting the person very quickly and were much more lethal.
Germany fell to the Allies in 1945. its chemical stockpile was seized, and the United States began producing its own variation of the German nerve agents, which became classified as G agents. The most widely produced was a far more lethal version of sarin nerve gas. But by the early 1950s, an even deadlier nerve agent was discovered.
A British company was investigating some insecticides and came across a particularly potent one. They looked at it and then referred it to the United States in about 1953-54 time frame. And the United States looked at it and realized that it was a whole new series of very potent nerve agents that had been discovered. They looked at all of them and then decided that probably VX was the one that they would like to go with.
From 1961 to 1968, the United States produced approximately 4,400 tons of VX, enough to kill every human being on the planet. The U.S. stockpile served as a deterrent against the Soviet Union, which had begun producing even larger supplies of chemical agents, including the nerve agents Soman, sarin, and VX.
In 1960-61, we standardized a artillery projectile, and we came up with a newly designed landmine, and also loaded VX in a rocket warhead.
Fortunately, neither country ever used a lethal nerve agent against the other. In 1969, Richard Nixon agreed to ban the U.S. manufacturing of chemical weapons. Production of VX ceased, and the stockpile was placed in storage. Today, only the United States and Russia claim to have stockpiles of VX. Yet some suspect that Iraq may have produced large quantities of the deadly nerve agent. At the onset of Operation Iraqi Freedom, U.S. forces prepared for the worst.
We felt that it was very likely that our adversaries would use chemical weapons against us, and we felt that they had VX in their arsenal. VX is often used as a terrain denial weapon, kind of the way you would use landmines. And one of the things we're very concerned about is not having our forces stumble into land that's been contaminated with VX.
The main goal at Edgewood Chemical and Biological Center in Maryland is to provide protection and detection for the U.S. troops in Iraq.
We have the best protective force in the world, and each warfighter has all the protective equipment that he or she needs, the protective clothing and the protective mask. The masks will totally protect you against inhaling VX, and the chemical protective clothing is specially designed so that the VX can't penetrate it.
In January of 2005, the United States ended its hunt for weapons of mass destruction in Iraq. No VX was found. But the threat of VX doesn't end in Iraq. Other countries are suspected to be attempting to produce the deadly agent. And there is enough VX in the US and Russian stockpiles to kill millions. So the threat of attack from one of the world's deadliest weapons remains.
Okay, so why am I showing you all this stuff about VX gas and nerve gas? It doesn't make sense. Well, we can really look at the effects of neurotransmission by looking at something as potent as VX gas. What VX gas does is it inhibits the enzyme that breaks down acetylcholine in the synapse. So you have this huge flood of acetylcholine, and you'll like start having violent contractions, and you'll be like spasming, which then leads to paralysis, and you'll stop breathing. It's terrible. I mean, it's kind of scary. Botulism does the opposite thing. It blocks the receptor sites for acetylcholine, so you have paralysis that way. It's kind of fascinating that we can look at one neurotransmitter and we can talk about Alzheimer's disease, food poisoning, and even chemical agents. So let's go to another neurotransmitter. Oh, what's going on here? Here, we want to talk about dopamine. Dopamine is a neurotransmitter that's involved with the initiation of motor movement. It plays a huge part in attention, learning, and memory. What dopamine also does, and again, there's lots of stuff that these neurotransmitters do, but I'm just giving you a brief introduction. Every single drug of addiction increases dopamine in an area of the brain called the mesolimbic system, which is the central reward pathway of the brain. So, we have drugs like Ritalin, even cocaine. Cocaine blocks the reuptake of dopamine, so you have a lot of dopamine in the synapse. Serotonin is another neurotransmitter that I bet you've heard of. Serotonin is involved in the control of the sleep-wake cycle, your mood, and your appetite. An interesting thing, though, is that most of the serotonin in your body is not as you would think in your brain, but it's actually in your gut, like in your stomach, in your intestines, really. So a lot of times you'll see commercials about drugs for serotonin that involve gastrointestinal distress. That's why. That's one of the reasons why. So deficiencies in serotonin have been associated with sleep disorders, aggression, and depression. And drugs that increase serotonin, like Prozac, have been used for a long time to treat depression and anxiety as well. Endorphins are a class of neurotransmitter that inhibit transmission of pain information. I'll bet you've heard of people abusing opiates. So morphine, heroin, hydrocodone, oxycodone all act by facilitating endorphin transmission. So the word endo means within, and then phene is like morphine. So endogenous morphine. We have morphine within us. We have a natural substance. But we can also take drugs to affect that area. Glutamate is a universal excitatory neurotransmitter. It's an amino acid that's derived from glucose and plays an important role in excitatory functioning of the neurons. Maybe you've heard of MSG, or monosodium glutamate. It's a flavor enhancer, but it's also a neurotransmitter. Gamma aminobutyric acid, the universal inhibitory neurotransmitter, is found in the brain and the spinal cord and plays a number of roles, but it's super important in regulating arousal and anxiety. So a lot of people take medications that increase levels of GABA, like Xanax or Klonopin or Ativan or Valium, to treat anxiety. So that's a few of the neurotransmitters. There's a couple other things that I wanted to show you here. Let's zoom out a little bit. I want to talk about the central nervous system, the peripheral nervous system, the sensory division and the motor division, which are very important in the peripheral nervous system. Remember the three types of neurons that we have: sensory and motor. Well, in the motor division, there's further divisions. There's the autonomic nervous system and the somatic nervous system. When we talk about those, the autonomic nervous system controls like smooth muscles. So like heartbeat, respiration, stuff like that you have no control over. The somatic nervous system controls skeletal muscles or striated muscles. There's a couple divisions of the autonomic nervous system: the parasympathetic division and the sympathetic division. Both of these are very, very important, and you need to know the difference. We'll start with the sympathetic nervous system. I'll bet you've heard the term fight or flight. A lot of students think that that's the same thing. Well, fight or flight is not one of these and this one. Fight or flight is all sympathetic, because you need the same thing to run away or to fight off something in your environment that is threatening you. And the parasympathetic division is the opposite. So, go to your book and look at this chart right here, and it'll show you what the parasympathetic and the sympathetic nervous system control. So remember, think fight or flight for sympathetic. It's sympathetic to the grizzly bear that you stumble upon in the woods. Well, what do you want to either fight or run away? What do you want to automatically happen in your body? Well, look at the red here for sympathetic. You want your pupils to dilate so you can see stuff up close. You don't really need to be drooling because it's not like you're going to be snacking on any food. Your breathing, you want to speed up your breathing because you need more oxygen. You want your heart rate to increase, and that's what the sympathetic nervous system does. It's not really time to digest your food, so it stops digestion. It releases glucose from the liver, which is like the fuel for your muscles. It relaxes the bladder, and it can actually stimulate the sex organs. So, like in males, that would be like ejaculation. Now, the parasympathetic nervous system on the other side, think of rest and digest. Okay, so it constricts your pupils to little pinpoints so you can see danger on the horizon. It stimulates saliva so you can help digest your food. It slows down breathing and heart rate. It increases digestion. And it contracts the bladder. And it also stimulates sex organs. The best example for this is men because they're a lot easier. So in men, it would stimulate an erection. We'll talk about that in later chapters. But for now, that gives you a good idea as to what's going on with neurotransmitters, and we've had a brief introduction to the parasympathetic and sympathetic nervous system. So keep reading your book, participate in the discussions. You got a lot to learn in this chapter, so take your time, spend extra time with reading, and as always, ask me if you have any questions.